Today is about vision, which is a lot more than the eyes. And basically the eyes obviously provide some the basic input, but your brain is the thing that's interpreting it and that goes way beyond that. And the word that's often used to describe this is that it's a constructive process, that it takes the kind of the input or the raw building blocks and it constructs something beyond the information given. And the vision is a good example of this, but it's something that our brain does in general. So in the social world, for instance, we go way beyond the information that's given there too, to make inferences and so on. So here the example of the Kinesia triangle is that we have the sense of there being something like a white triangle here that isn't there. And in fact, it's very hard to see that as being kind of three corners without there. So our brain assumes that there is a surface here that's doing that. And there are a set of principles or assumptions that the brain follows, some of which might be innate, some of which might be learnt, that basically one object on top of another occludes it, for instance, blocks it out. But the brain is going far more than the information of the eyes. So in terms of how the information goes from the eye to the brain, there are multiple routes that do this, but the main one that gives rise to our kind of rich and detailed and our conscious visual experience goes to the primary visual cortex. And it's called the primary visual cortex because it's the first cortical part of the brain to receive inputs. There are non-cortical parts on the way up. And your primary visual cortex is in the back of the brain, not near your eyes, exactly the opposite end, in between your two hemispheres here. And one of the things that confuses the hell out of people is that basically how the visual world is kind of segregated into left and right isn't quite the same thing as what happens in your eyes. So basically an object on the left side of space, you can see it in both of your left and your right eye. And this is one of the myths that people have, is that they assume that something on the left only goes to your left eye, but you can easily prove that that isn't the case. It goes to both eyes. But so here this yellow object activates this eye and that eye, but the image falls on this side of the eye here. But then what happens is that the side of the eye goes to this side of the brain. So basically this side of the world goes to this side of my brain, but it doesn't go into one of my eyes, it goes into both of my eyes. But it goes primarily onto one side of my eye. Okay? So the left side of space goes to your right brain, and the right side of space goes to your left brain, but it's not that the left side of space goes to your right eye. That is not how it works. Okay? So don't make that mistake. And there are a number of processing stations on the way that kind of help to elaborate the information from the eye. The retina of the eye itself also performs some kind of computations on the visual input as well. So this, what's called the geniculostriate pathway, is the main pathway to the brain. So the way station on the way is called here the lateral geniculate nucleus, and this contains neurons that respond to light stimulation. But remember, at this point, it's not responding to light. There is no light in the center of your brain. All there is the firing of nerves. Okay, but it's doing that. And the striate cortex is another name for your primary visual cortex, also known as V1. So you've got these three names. Striate means kind of stripe, and in the old days, when you stained brains using certain chemicals, you would see a kind of black stripe that went through it because of particular cells in that part of the brain. So on the way from the eye to the brain, there are, again, way stations that help to process information. So your lateral geniculate nucleus are cell bodies that carry information from the eye to the brain that have this particular property. So a receptive field basically is just the area of space that a neuron responds to. So some neurons might respond to light here, some neurons might respond to light there, other neurons respond to light here, and so on. And within that region that they respond to with light, they might have particular characteristics. So it might respond just to light in the center, but not in the surround. And that's kind of what you have here. So what the circle represents is a region of space, and you shine a light in there and you see how the neuron fires. So when you shine a light on this bit of space, here the neuron fires. So when the light's on, ping, 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 ping. So each one of these lines is the firing of a neuron, and this is time. When you take the light away, it switches off. If you shine a light on the bit outside it, it doesn't fire. But when you take the light away, it does fire. Okay? So this is slightly puzzling. So what you imagine is that neurons only respond to when a light is on, but neurons also respond when the light is taken off as well. So in a way, we often like to think of the brain as kind of a little bit like the pixels on a TV screen, but this is nothing really like the pixels on a TV screen. So it doesn't just respond to light there, it has kind of edges, that it responds to light only in the middle, but not light outside. It actually switches, it fires when the light is switched off in some cases as well as on. And you can have ones that show the opposite properties. So this one here is effectively kind of coding dark in the center. So when a light is in the center, it gets switched off, and then when you kind of release it, it comes on. So that responds to the absence of light, and then here. So although these are responding to relatively small amounts of space, you can see that they are coding something complex. And one reason why it might have these kind of on-off properties is that it's good for detecting kind of edges or boundaries where one thing begins and one thing ends. It's a way of kind of segmenting things up. And we'll come back to that later. But that's the essential property of how neurons fire when information is presented to the eye. You've got the lateral geniculate cortex. And then your primary visual cortex, you have neurons that respond to more complex information. So some neurons in the lateral geniculate nucleus would also respond to colour. Your primary visual cortex also has neurons that respond to colour. But rather than responding just to points of light, it responds to like bars of light, which are like edges, to lengths and so on. And this is the classic kind of early studies where you would put an electrode into a living brain, say cats and monkeys, and record from them. And this was what Hubel and Wiesel did. And this effectively is kind of the origins of the kind of the grandmother story, a grandmother neuron story that I said in the first lecture. So the idea is that you might have some neurons that respond to kind of points of light, maybe this on-off way. Then you have some neurons that respond to strips of light or kind of edges where light and dark kind of abuts. Then you've got neurons that respond to surfaces, neurons that respond to shapes, neurons that respond to whole people, and so on. Is that the idea that vision is hierarchical, and who knows what's at the top of the hierarchy. But that's the basic idea here, is that we've got building blocks from the bottom upwards. So what you have in primary visual cortex is that you might have cells that respond to, say, a bar of light, so an oriented length of light that's on here, and also if you shine light in different parts of this here. And this is what's called a simple cell, a neuron that responds to light in particular orientations, either along its length or a part of its length. The way we could think about that in terms of a hierarchy is in terms of building blocks. So if you've got one of these neurons in lateral geniculate nucleus that responds to a centre here, another neuron that responds to a centre there, and another neuron that responds to a centre here, you can kind of almost build up a diagonal line, and it's kind of saying, I'm looking across all these 4 or 5 neurons, and I'm seeing what's common across them. And that's kind of what's represented here, is that this is a neuron in your early visual cortex that's receiving input from these kind of on-off the neurons here. And by doing that, you can almost build up the little kind of elements into something that is more like a line, or something that's more like an edge, something that's more complex than the inputs with it. So simple cells we could think of as being combinations of these center surround cells that are present in the earlier brain region in your lateral geniculate nucleus. So they respond to different orientations, but they also respond to kind of points of light along this. Complex cells are kind of seen as being the next stage in the hierarchy, where you can imagine that several simple cells are being combined. And here what you would have, the characteristic is that it responds again to lengths a particular orientation, so it might respond to this orientation and not that one. But what it might also do is that it might respond to particular kinds of lengths. For instance, you might have to stimulate the whole length of the receptor field, not just a part of it, to get it to fire. Okay. And then you've got hypercomplex cells, which are combinations of these as well, which might effectively be lines that of a certain length that have stoppers at the end. So you could imagine kind of light on going in one direction, then light off kind of providing stoppers there. And the idea is that you can kind of build vision up in this particular way. So one of the properties of the primary visual cortex is that it's spatially organized, and it kind of reflects the kind of spatial organization of the input on the retina. So the input on the retina has a number of properties. One is that the image is upside down, and that's just what you get by putting an image through a lens, is that it kind of rotates it. But also in the back of the brain, the bottom part of your primary visual cortex represents the top part of space. Okay? And then you've got this left-right division, that the left of your primary visual cortex is responding to the right. So it's almost as if it contains an image that is left-right, reversed and upside down. But of course it is not an image. There is no image in the back of your brain. All it is the firing of neurons that are responding to things. There is no light there. There is no image. You might kind of wonder, well, where in the brain turns the kind of image the right way round? And the answer is nowhere in the brain does this. It doesn't need to. And that kind of presupposes that there is a right or wrong way for things to be up and down. All you need to know is that when something is activating the lower part of my visual cortex, I need to reach upwards and not downwards. So you can just calibrate your kind of reaching movements to do that. You don't need to turn anything the right way around. You just need to know when this part of the brain is active, it's responding, it refers to something up there. That's all you need to know. You don't need to do any kind of correction. What this also means is that if you damage your primary visual cortex, that you would be cortically blind. You would not be able to see a part of space. But unless you damage both of your primary visual cortex, you might be able to see some parts of space and not others. So in this example here, a patient has damaged their primary visual cortex in one hemisphere, and what they would then have is that they would be blind in one side of space. And that would be true irrespective of whether their left eye was open or the right eye was open. What would that be like? Well, what you would naively imagine is that you would be able to see things here and the rest of space would be dark. But that isn't the case. What you would have is that you would be able to see things there and the rest of the space would not exist. So I don't know about other people in the room, but for me, vision kind of ends here, but I don't have darkness behind me. I just have nothing. There is nothing there. And that's exactly what it would be like to lose half of your visual field. You would have, that would be your vision, and the rest would be nothing. It would not be night, it would not be dark, it would be nothing.
So that if they continue turning their heads, then I'll be able to see that?
Yes, so as they turn their heads, in the same way as this here that I wouldn't see, I would then be able to see it, but you would lose that. So it's like having a narrower thing. Yes, so they would be able to do that. Yeah, exactly. And similarly with the eyes as well. If they move their eyes, things would then fall into it. That's right. So you can compensate quite well for it. So tunnel vision is actually where you damage your retina and you just have this. But you could imagine it as being like a particular form of tunnel vision. If you damage an even smaller part, so if you damage the bottom bit of your visual cortex in one, you will lose a kind of a quarter of your visual field. That is very hard to imagine what that would be like as well, or little parts of it. Again, when you lose a part of it, but if you've got information around it, you might end up filling it in. So at the moment, we have at the back of our eyes what's known as a blind spot, where there is no visual information. But again, we don't see a black hole in our visual field. We kind of fill it in. And to some extent, when you've got these partial deficits, you would do that. And in the same way as now, I probably have a sense that there is bricks and things here, but I'm not sure I literally see them.
I thought that our blind spots were compensated by the visual field of the other eye.
Yes, they are. Yeah, that's right. But you still wouldn't see it with one eye closed as well. You don't see a black hole when you do it. But yes, you are right as well. Yeah, that's a really good point. Yeah, so because of the different alignments you do. There are ways of doing it that you can bring objects in and you can make them kind of disappear. You have to keep focused on one thing, and you can download those kinds of tricks online. So that is what it is like to damage that. So the other thing that's also puzzling about this is that this is the route that I talked about from the retina to the lateral geniculate nucleus, the primary visual cortex. I won't go through all these little minor divisions here. Is that basically there are multiple other routes to seeing that go from the retina. The reason why this exists is partly just to do with evolution, is that in effect the primate visual cortex is kind of added on top of earlier kind of visual cortex that more comparable birds and reptiles and so on. So we don't kind of undo earlier stages of visual processing, we've kind of added on new machinery. So it kind of means that we have a kind of a bird brain vision as well as a primate vision as well as a kind of reptilian vision all within the same brain would be one kind of rather simple way of putting it. What that also means is that if you were to damage your primary visual cortex, you would be cortically blind. The doctor would say you were registered blind and so on. But what it would mean is that if you put a bit of X here, all these other routes would still be functioning to some extent. Now this is quite puzzling because if these other routes are functioning, why is it that you can't see? And the idea is that this route here is particularly important for kind of more conscious vision and the kind of the detailed vision that we have. But what that means is that these patients would have unconscious vision for things that they are blind to, in effect. And that's the phenomenon called Blindsight. It also means that they would have other phenomena as well. So there are, let me talk you through some of these. So this one here is related to your biological clock. And basically this receives light information from the retina to tell you whether it's night or day. If you go on a flight to New York and so on, it's the information that tells you it's light and so on that helps you to come get over jet lag. If you've got a patient who's cortically blind, they will say, I can't see things. You can put them on a plane to New York and their body clock will reset using light information with that, because that is a different visual route in their brain. Okay, and they are still using visual information. Perhaps more bizarrely, and this is a phenomenon that we would all have, is that these regions here are kind of more evolutionary ancient, and these are involved in kind of orienting the eyes to different stimuli. But we've all kind of had it that maybe, for instance, we've seen something important, a friend or something, in the corner of our vision, and we've turned round to look, and then we've realised why we've looked. Now, the thing there is that why did we turn to look at our friends if we didn't see them before we moved our eyes? Okay? So it's like we knew they were there, but we didn't know they were there until we moved our eyes. And the paradox there is because there are different visual routes, one that tells you that there's something important there, and then one that tells you consciously what it is. And that's why we can orient our eyes to something, we don't realise why we've done it afterwards, because there's one route that tells you there's something over here you need to look at, and then you realise what it is. And to some extent, patients who have cortical damage, whoever asked about the things, would also be able to do that. So despite being, in some senses, blind about things, they might still be able to move their eyes into that region if something is flashed, which is very paradoxical. So the experiment that's being done here is that basically he's looking straight ahead and there is something in his blind field that's moving like that. If you ask him, can you see it? He says no. That's what he says by unaware. He says, I cannot see something there. Then the experimenter says, that thing that you can't see, is it moving to the left or the right? And he gives the correct answer 90% of the time. Okay? And that is, in effect, the phenomenon that's described. So he says, I'm not aware of anything moving here. I'm going to say the word left. Okay? And he's correct with that. So how does he do that? Well, the reason he does it is that although he has damage here in this particular, well, here actually in the primary visual cortex there, There are other routes that do this. So this one here is also involved in motion, but there are others that enable you to process where things are, say in your periphery, that might be helpful for orienting your eyes. Here you can see he wasn't moving his eyes to left or right. He was told to do that. But there's nevertheless that kind of information that could orient your eyes one to the side. So basically what this suggests is that this particular route here is particularly involved in conscious vision, those particular aspects. What that means for other animals who lack this, whether they are simply unconscious of their visual, I do not know. But that would be one way of thinking about it. So this is the phenomenon of blindsight here. And basically, again, to some extent, these patients would fill in their kind of blind spots in the same way as we would fill in our blind spot, either from information from the other eye, if one eye's closed, just from the surrounding information of texture and colour, and so on. So these are the kinds of experiments that were done where you show, so the black region here is, say, the person's blind spot, but maybe they've damaged this bit of the visual field. If you show them, for instance, a semicircle, here they would say that's a semicircle. But if you have the semicircle abutting their kind of their loss of vision, then they have a sense of it being filled in, and they describe it as a circle. Now, the reason that they do that is that the information is being passed up the hierarchy, and there are things that are kind of looking at the information there and saying, yeah, I'm going to fill that in. This is a bit like your Canisa triangle. Remember the triangle at the front, where you're creating surfaces from partial information, and that's kind of what is happening here, is that stages above the primary visual cortex are making a decision that there's a circle without there being anything there. And of course, if it's presented completely in the blind field, he doesn't see it at all, although he might be above chance at telling you it's a curved object, for instance, if you were to force him to guess. Any questions about blindsight? Okay, so it's a very kind of interesting phenomenon, and basically the essence of it is that it's damaged your early visual cortex, and you can still do it because there are multiple routes from the eyes to the brain, of which this is only one. So this is your visual cortex here, and then when you're in your primary visual cortex, again it branches out. So here you've got like one branching structure from the eye to the brain, then when you're in the brain you've got other branching structures out from this primary visual cortex. So it's a bit like a hub or a way station, and then beyond that you've got things that are specialized, and again things that are specialized then for color, motion, shape, other kinds of information that's important for our visual world. And that's what all these other visual regions is here beyond V1, that they're coding more specialist information. But again, what you would have is kind of an element of divide and conquer, is that you have some specialization within these regions. So the function of this region here is different from the function of that region, even though both are responding to visual information, that they are specialized in terms of doing. Yeah.
When it comes to these sort of like large branching networks and stuff, for the MCQ, I'm assuming you don't expect us to.
No, I was, yes, I've been a little bit worried about MCQ. I've read them, but I don't think they're that bad. Yeah. But absolutely not. So I would never give you a question, for instance, well, you could probably guess it if I got rid of that one and a question mark and put in that. I would not do that. I might ask a question such as, what are the characteristics of damage to the primary visual cortex? And one answer would be, you can't see in colour, you claim not to see things, but you're better than chance, or something like this. And that would be a typical question. Yeah, exactly. Or I might say, what is the name of it? And then I'll say, is it blindsight, achinotopsia, and so on. Those are kinds of the levels of the questions. So when I talk about some of the key regions, if I talk about a region over and over again, there's a chance it may come up. But if the things are just popping up, I wouldn't say. Yeah, exactly. But yeah, I have been aware that you would be panicking about this.
On that note, is there any resources that we can use to test ourselves before the test?
Yes, I will circulate them by email. They are a little bit buried on the website. They are up there, but I will send them around to give you some clues. When I send them around, though, it might cover some of the things that aren't covered in the lecture, and don't let that trip you up as well, because it's just an example of the questions I will make sure that they are covered by what's in the lecture. Yeah.
It's more to do with actual blindness.
Sure.
When you've got one area of V1 actually damaged, do you see half and half? So say I'm looking at you, only see half of your part, I can see double of you.
Yeah, I think if you're kind of in the midline, my understanding is that you would kind of almost be filled in. I don't really know, because it's a bit different from us, because when we fill in stuff at the edge, it's because the information is kind of vague. Yeah, I don't know how detailed it actually is, basically. But they, in the same way as I don't have a sense of my vision just suddenly ending, it just kind of seems to fade out. I can only assume it is like that.
Because I was thinking, obviously, each eye's got two visual spaces.
Yes.
So it'll be half and half for each. So if it's left of each,
Yes, that's right, yeah.
you see half of each image.
Yes, that's right, yeah, yeah. So you would still have stereo vision. You would still have stereo vision. So the two regions I'm going to talk about are more specialized visual regions. One involved in color, which is called V4, and one involved in motion, which is called V5 or NT. And basically the original kind of brain imaging studies that looked at this, so PET was just a forerunner of fMRI. What they did is they looked at coloured blobs. There's an artist called Mondrian who basically has lots of coloured squares, kind of abstract artist. And they present kind of coloured squares versus grayscale squares. And what you find when you do that is that obviously both colour and grayscale is activating your V1. So those kind of cancel out when you compare them. But there is a region that is more interested in colour than interested in grayscale, and that is this area called V4, your kind of fourth visual area. Okay. In another condition in the same study, what they had is an array of dots, so loads of dots on a computer screen. In one condition, the dots would move. In another condition, the dots were static. When you do that, you don't get your area V4, you get another area called V5. And actually, even before this, these areas were postulated to exist in humans because similar things had been found in the primate brain. So this wasn't the original discovery, it was one of the first demonstrations in vivo in humans. But the specialization between colour and motion was known before that. And as with all of these regions of the brain, you have two of them, one in your left and one in your right. And as with your early visual cortex, your primary visual cortex, they are specialized for different regions of space. So my left V4 is involved for seeing colors in my right side of space and so on. My left V5 is involved in seeing motion in the right side of space. So the one in red here is the motion sensitive area, and in blue it is the color sensitive area. It's slightly puzzling to think why there would be specialized brain regions for color and motion. And the reason for that is that your retina is also sensitive to color and motion. So why is it that the brain needs to dedicate a large, well, relatively large area to processing these things, given that there is coding of that information at much earlier stages? And the answer to this is that basically the colours are not exactly what you see. And what I mean by that is that if you look at one object in one lighting condition, so if I look at a banana in this room, take it out and so on, it will look the same to me, but actually if you measure the wavelength reflected off, it will have different wavelengths coming off, because the light from the sun is different from the light from here, and so on. And this is the phenomenon known as colour constancy. Basically, what you're trying to do is you're trying to figure out what the true colour of an object is, irrespective of what a light source is, whether the object is in shadow or brightly lit, and so on. And the best example I can give of this is the dress. So I cannot believe that anybody in the room perceives that as gold and white, but I bet that they do. Is that right? Does anybody see? Yeah. Who perceives that as gold and white? Okay. Yeah. Who perceives it as black and blue? So for me, this is quite extraordinary. And is there anybody who can change their percept? That you can make it what you want it to be? Yeah, it's okay. A few people can. It's quite unusual. I don't believe it, but I'm told people can. This is colour constantly, and basically what's happening here is that everybody's retina is being stimulated in exactly the same way. Okay, here. So let's get rid of that information. what's happening is that our brains are interpreting it differently. And it's partly nothing to do with the dress, it's partly because we don't know exactly what the lighting condition is here because of the cropping of it. And the best way I can describe it is that this is how it is in Wikipedia. So if your eyes look from this patch to that patch, you can convince yourself it is the same colour. And this little bar in between proves that it is the same colour between there and here and here. It's exactly the same colour, physical colour that is, even though you perceive it differently. So when you've got the cross cropping, you're trying to figure out what is the true colour based on whether it's a bright light. So this is black and blue in a bright light, and this is white and gold in dark. So in effect, whether or not you see this as white, as gold or black, depends on whether you're interpreting the light source as being bright or dark here. And depending on that interpretation will affect how you see it. So that's the example of colour constancy. And basically what you're trying to do is figure out what is the true colour irrespective of the lighting. So you're doing something more complex. In order to do that, you need what are called large receptive fields, so not like the little regions of space that you see in the lateral nucleus. You're trying to figure out what is the light source in this room. So you have large receptive fields that enable you to kind of compare individual points with kind of what is the light from above. In this close cropping, you can't quite figure out what the light source is, and this is what's throwing. Okay, so that's what V4 does, is that it's trying to go beyond the information given here by colour-sensitive cones in the retina and try to figure out what is the true colour of the surface, irrespective of this. What's also, yeah.
So just to summarize, it's sort of attempting to filter out the change in light pattern.
Correct. Yes, it's a filter. Yeah, it's a filter. It's filtering out the background light and trying to figure out what the true colour is. Correct. Yeah, exactly. Yeah. If you damage your V4, basically what happens is that you see the world in black and white. And this is called achromatopsia or cerebral achromatopsia. It is not the same as colour blindness. So people who are colourblind because they lack a particular colour receptor on their retina, they still report seeing the world as being colourful, but they just, on formal tests, get them confused. They might call that green instead of red and so on, but they still see the world as colourful. Patients with damage to this part of the brain see the colours as either missing or washed out. If you damage half of your V4, this side of the world is colourful, this side is kind of washed out, and you kind of have a bit of a gradient in between where you fill in the information. It doesn't suddenly end because you can kind of figure out from the blue that the blue goes on, and eventually over here it will be properly grey in effect. So you have these kind of filling in effects as well. What's also interesting is that you can kind of show the patients who have damage to their the V4 that they can still detect wavelengths of light in the retina and so on. Or even, for instance, if you have a green and a red that are next to each other, so you've got a green square and a red square that are abutting, and you say, you know, can you tell these colours apart, which one is which, and they can't, but they can still, the cells in their early parts of the brain can still tell that there is a chromatic difference there. And the reason for that is that there will be cells that are kind of colour sensitive that can detect little kind of edges like this that can do that, but they don't give rise to a conscious vision. So some of these might be colour sensitive. So the patient can still make some colour discriminations despite claiming to not see colour. But they can't tell you what it is, and they don't have a colour experience. And they can only do it if two colours are next to each other, because the cells early on have small receptive fields. They deal with small parts of space, not big parts of space. Okay. So that's V4. V5 is involved in motion perception. And basically, if you damage your V5, you can see in colours, but you see the world as snapshots. You do not see smooth transition between objects moving in space. It's like a series of still frames, and that's what's described. So this is somebody who's said to be achinotopsic. And basically, it would be a series of snapshots would be one way of describing it, but some of the other things have been said. If you pour water from kind of a jug, that people describe it as seeing the water almost frozen. It looks like it's kind of a solid thing rather than a running stream, like a frozen waterfall type of thing is one of the things I kind of remember as a poignant thing. If you go into kind of primates and kind of look at the specialization of cells in this region, you find that they don't respond to colour, but most of them respond to movement. And again, neurons are tuned to different directions. So one neuron might respond to movement in this direction, one to movement in that direction, and so on. What was also mentioned in the videos that you would detect movement in other sensors. That's one of the reasons why it's probably a little bit frightening. So as you're going down the escalator, you would be able to feel that, the thing going through your hands, the railing, or the sense of motion as you go down the escalator. Or similarly, when the train is coming, you would hear the whooshing, the kind of Doppler-like effect as it comes. So it is a specific visual deficit that you have, and specific to visual motion. So as you kind of go beyond this, again, what we're interested in next is kind of object recognition. So far we're just talking about abstract properties like shape, or not shape, so motion, colour, and so on. What about actually recognizing actual objects? I mean, for this, what we're particularly interested in is kind of linking vision really with memory. So objects exist because we have in our head, we know what a cup and a pen is, and we have kind of stores of what these objects are, what they look like, and so on. So we're at that point, we're going beyond information, we're linking it with stored knowledge of people, animals, this sort of thing. And in effect, what's going on here is that object recognition is involved in the ventral stream, and that is the kind of thing I might ask in a multiple choice, is that the ventral stream is kind of for object recognition, the dorsal stream is for kind of acting on objects. But a simple model of object recognition is something along the lines of this, that you've got an object that is then decomposed into multiple parts according to these specialized regions. So you have separate regions for motion, color. But the thing that's probably most important for recognizing objects is its shape, which is here is the word form. Depth is obviously important, mainly for reaching and so on, rather than necessarily recognizing. What you then have is a whole set of kind of more advanced perceptual processes where you're grouping different edges together, grouping different surfaces. You're linking together different parts of the vision to say, right, those things belong together as separate from that. It's often called figure ground segmentation, is you're figuring out that that line and that line are part of the same object, for instance, with the others. And that's kind of what's shown in green here, and I will kind of go through them. What you have here in purple are what are in effect memory systems. So semantic memories, kind of your knowledge of objects, how they're used, their names coming out here. But the idea that we also have a store of objects. We have something in our head that tells us what the shape of a cup, an animal, a dog, and things is, and that is stored. It's not just there, constructed primarily from the retina. We actually store that. So these are kind of storage systems. And basically, if you have damage to one of these regions, you will have problems in recognizing objects from vision. And the term for that is agnosia. So we've had blindsight, which is kind of very early processing, achinotopsia, achromatopsia here, and then agnosia is problems in knowing what an object is, so being able to distinguish a cat from a dog or a table from a chair, this sort of thing. So basically, to create kind of visual descriptions of objects, what you're having to do is to group smaller elements together. So this is, again, part of the kind of hierarchical nature of vision. And these are the kinds of ways in which you kind of group objects together. They're called the Gestalt principles. So basically here, you know, people tend to perceive that as 3 rows instead of 4 columns. Okay, you could force yourself to think of it as 4 columns, but that is not the kind of the intuition. So your brain has biases to perceive the world one way more than another, and one idea behind the people with the dress is that some people are nighttime people, some people are day people, and that's your bias as to why you see it that way. But here, this would be a natural bias, that things that are close together are perceived as belonging together. So we have, in effect, 3 clusters here, rather than any other combination. Here, technically, you could imagine that these are 2 V-shapes that are kissing, but that's a slightly odd, counterintuitive way. Most people would interpret them as 2 lines that are crossing. And this is, again, a bias in our visual system to do that, and it's partly based on our experience of this. This is grouping by colour, so here we would think of those as being kind of columns rather than rows because they're similar. And here we might imagine that that's an occlusion, so your Kinesia triangle and so on. And all this is done in kind of slightly higher levels. So things like the Kinesia triangle, this white triangle that we had at the beginning, is beyond V1. You get it in these higher order visual cortices that then kind of infer that there is something on top of the other one. V1 doesn't care really about the Kinesia triangle, but higher order regions are kind of figuring out how all these different parts are going together. There is another region that is particularly interested in shapes, but doesn't really care whether they're real shapes or made-up shapes. And this, to some extent, would be similar to your colour and your motion specialisation, but here for shape. So this lateral occipital complex, LOC, responds in fMRI to seeing an object more than a texture, but it doesn't care whether it's a real or a made-up object. So again, it's quite a basic description of them. But it is a little bit more sophisticated than that. So it treats an object the same whether it's partially occluded or not, in effect, by which means you get the same pattern of bold response. So in effect, it's saying that this object is the same whether I kind of hold my hand over it or not. It's treating it that there is a whole object there, even if you don't see the whole object, part of it is being missing, or that there's another object in front of it. So it is doing something a little bit more sophisticated than just grouping things together.
Wait, can you just say that again?
So occlusion means that you've got one object in front of another, so it would be partially obscured. So what you might imagine is that if it treats an object as a whole. So if the brain was to respond to a whole cup, it would respond to half a cup or that. So it's assumed, the interpretation is that it's saying that the whole cup is still there, even when you only see half of it, I suppose would be the interpretation. Whereas other parts of the brain would take things at face value. They would say, oh right, it used to be a long line, now it's a short line. Okay, but this does not make that assumption. It fills in the missing parts. And if you do TMS over this region, it disrupts people's ability to say whether shape A and shape B are the same or different, for instance, in simple reaction time tests. So again, what we can say is that this is kind of involved in an early computation of what you might call shape constancy. So we've had colour constancy with the dress and so on. But here is that the shape is the constant shape, irrespective of whether I put my hand over it or my fingers and it disrupts the visual image of it, that you assume that there is a constant surface or object that is there. So we could think of this in terms of kind of putting some brain around this simple box and arrow model, that this lateral occipital complex involved in shape is kind of binding different features into a kind of grouping, but it's not here, it's not part of your knowledge base, because it doesn't care whether an object is a real object or a made-up object, something that you've never seen before. So it doesn't seem to look like a memory store as such. So he has agnosia. So if you were to look at his visual fields, by which I mean you kind of shine points of light and say, can you see something or not, he would be intact. So not blindsight or anything like this. He has kind of full 20/20 vision and so on. What he can't do is make sense of it, the information around him. It's very hard to imagine what that is. It's kind of like being able to see lines and things like that, but not being able to join the lines into faces and edges and so on. So here, his kind of disruption is being able to group parts into wholes. The reason we know that is that there are certain things that he can do. So for instance, in terms of what he can do, is that if you give him a line drawing like this, I think, is that his drawing or the one he was given? But either way, he's pretty good at doing that, so he will slavishly kind of copy the lines and get them in the right place. But if you ask him what he's just drawn, he might not be able to say it's a kind of a cityscape with a cathedral dome or whatever that is. So he can see all the different parts and be able to reproduce them on the page, but be confused about what that is. Originally, he was able to draw from memory. So this was a picture of an owl. So even though he wouldn't be able to see an owl or recognize his own thing of it, originally he could draw that, which suggests he had kind of an image of objects in his head that he could reproduce. Because he was one of these classic case studies who was tested over decades, they then went back and tested him, having been kind of agnosic for, I think it was 2 decades, and found that he'd lost this ability. They became a lot more schematic. And this suggests that being able to hold on to your kind of images of what objects look like, if you haven't got the normal kind of input processing, in effect your memory of what things look like from the inside down starts to degrade. You need that kind of refresh. So there's an interesting kind of story there that that is kind of a yes and no. What he can't do is that he's poor at kind of naming objects from pictures. If you show him like a line drawing, he will struggle to say what it is and so on, or telling you whether these are real or not objects. This is a camera with a candle with kind of a doorknob on it. He would struggle to do that. So, but he could probably copy the different parts. Yeah.
Sorry, just to be clear, he had, or at one point was not an integrative agnosiac. So that's how he knew he could draw the owl.
Yes, that's right, yeah. Yeah, so it's kind of described as seeing the parts but not the whole. So you imagine somewhere within your visual hierarchy, you're kind of linking together lines and edges into surfaces and so on. Obviously, when you see the video, it's kind of surprising what he can do. So things like the bicycle and so on. So again, it's a little bit puzzling. It says he recognises his wife from the shoes. So what is the information there that he's getting from? Is it the motion of the bicycle, for instance? So remembering that motion and shape are rather different. He would have clues to the size of objects. So if I give a line drawing of a bicycle, you have no idea what size it is. But if you're looking at something through your eyes, you will know from depth cues and so on what the size is. It's a different kind of game at that point that you're doing. But clearly he can do certain things with that. It's not just that he can't see faces, he has problems with other things, text and things like that as well. So the idea with him is that he loses the ability to link different visual features together, and therefore he cannot access his knowledge of objects. But he is very articulate. He can describe what a bicycle is, that it has 2 wheels and so on. And to some degree he can imagine that, but obviously that has faded as well visually. In effect, what's happening here is that he's kind of losing his ability to kind of construct objects. In order to kind of go down here, you've got what was called kind of object constancy. And the idea here is that the idea that you can recognise objects from different viewpoints and so on. And obviously if you have this kind of damage there, you wouldn't be able to do that. In the normal brain, what's generally assumed is that you kind of have a memory store of objects, and that's how you obtain object constancy. So one theory is that if you've got something like a memory representation of, say, a cow, that you don't store every single instance of a cow, you store one particular thing of it, and then you match your input from your early processors onto that. So it's like a matching exercise that you're doing. And that's certainly one idea of a view invariance. You might store, for instance, the image of a cow in your head as being oriented this way. So when you see a cow facing you, you have to do a kind of mental rotation to get it there. And that's just a bit of a computational thing, is that given that I can see cows and faces from an infinite array of views, and I haven't got an infinite number of neurons in my head, how am I going to solve that? And one solution is that you don't store every particular instance, you just store particular aspects of it, and then you do a matching exercise with that. So that would be, here, the view dependent is that you would kind of have a mental rotation of objects. Assuming you can group the different parts together, you then have to match it onto the store. And basically this kind of ventral stream or inferior temporal cortex going outside of the visual cortex seems to be important. Here we've got the lateral occipital cortex. Your parietal lobes are probably involved more in kind of matching by views. So what is the evidence that these particular regions in the brain kind of are involved in object constancy? The kind of evidence, if you look in, say, monkeys and look at the properties of individual neurons there in terms of what they respond to, is that they have very large receptive fields. And this, but they respond to things like junctions, so things like a T-junction or shapes and things like that. But they don't care whether the T-junction is here or over there or whatever. So again, here you're losing at that point the left and right visual fields. It's just about there is an object in front of you. And the reason why that's important for object constancy is that the same objects are the same whether I have it here or whatever. It's exactly the same object. So there are neurons that are in effect carrying that property. It's the same thing no matter where it is. It doesn't change. Okay. What you can do in humans is that you can present the same object twice, say in fMRI. And if you present the same thing twice, typically what happens is that you get an adaptation, and that means that the second response is kind of lower than the first one. And you can do this with different kinds of objects and see how the brain is tuned into different object properties. And that's kind of what you've got here. So if I just kind of go through the stimuli, imagine that I present this one to you first in a scanner, then this one the second time. Here, this is exactly the same object and exactly the same visual image. But here we've got two different visual images, but of exactly the same object, just differing in size. Okay? So these are the same physically. These are the same not physically, but the same in terms of shape. These are not the. These are only the same in terms of shape if you rotate one to the other. Okay? But in terms of a physical image, they are not the same. Okay? Here, these, you have to know that that's what's called a telephone. They're not quite what looks like what's in your pocket, but the idea here is that these then are looking even more different from each other, and these are genuinely different here. These are not like. And in effect, you've got three different brain regions here that are tuned to three different aspects of object constancy. And what we've got here is that this is how much the brain responds at time 1 and how much it responds at time 2. And basically, if it treats them as the same, it will have a lower response at the second time. And you can see that all three of these brain regions treat that as being the same thing. Here for the lateral occipital complex here is that all three, this also treats it as being the same thing. But as soon as you've got a mental rotation here, this brain region can't tell whether that is the same or not. But there is another region that can, and that's further along in the hierarchy. So this region here recognizes that that is the same as that. So this is computing object constancy. This one kind of is, but in a more superficial way. This one is in a deeper way. Okay? Broca's area is involved in language. And basically, when you come to objects like this, that aren't just mere kind of rotations of them, you kind of then need to understand kind of semantically or linguistically that they're the same. And basically, your visual system cannot tell that these are the same object but your language system can, in effect. So at this point, you've left your visual cortex and you're in higher orders areas of the brain. But again, you can imagine that this is a form of object constancy, being able to label things linguistically as categorically as the same. It's just a non-visual form of object constancy. So again, here within the brain, you've kind of got this hierarchy of information as to whether things respond to shape, whether they respond to exactly the same viewpoint, and whether they respond to things as being categorically the same. And that might, the latter one might be more kind of language-based. But there is evidence that we do some kind of mental rotation when we're looking at objects or matching them. So this is the kind of task that they would give to certain brain damage patients, where they have to say which of the images on the bottom goes with that one. Okay? And here, in order to do that task successfully, you can't rely on the superficial aspects of them. So that looks a bit similar to this looks a bit similar to that. You've got to understand what the object is like in 3 dimensions, okay, to be able to do that. And again, there are some patients who have particular problems in that, in this kind of task. And they might be able to recognize objects when they're presented in a typical view, but not when they're in an atypical view, which suggests that they can match them either on the basis of features or other things, but they cannot do this kind of transformation. And that's why you've kind of got this other one here, is that there might be a separate route that's involved in kind of rotating images into a standard space, enabling you to recognize them. Okay. So here you've got grouping by shape. Here you've got what I call view normalization, so being able to kind of map things across that. Here, these are brain regions that, for instance, they can compute that a chair is the same when you kind of rotate it around. And then here you've got things that are involved in language that tell you that 2 very different telephones are exactly the same kind of concept, even though they look visually very different, for instance. And the way in which we kind of describe this is in terms of the what stream, or the ventral stream. And basically this runs from your visual cortex down your temporal lobes. So your temporal lobes are involved in high-level vision, but they're also involved in kind of perceptual memory, in language, in kind of conceptual knowledge. And you can kind of understand why this part of the brain is specialized for that, because again, they care about what objects are, but they don't care about where they are in space and so on. In the next lecture, I'll talk about what goes on in this other route in terms of attention and how we can kind of reach for objects and so on. In order to do that, of course, we need to know exactly where things are and less so what things are. So it's a different kinds of information is needed. Any kind of questions about that? I think just as a bit of a summary, again, what we have are different routes from the eye to the brain, which the main route terminates in your primary visual cortex. Your primary visual cortex is like a hub, and from that you've got another kind of radiation of different routes that have at least a relatively high degree of specialization for color, shape, motion, and then going beyond that into your temporal lobes here, you've then got more stored knowledge of what objects look like, and you're able to match vision onto those in order to understand them and kind of integrate the parts into wholes, recognize your grandmother, and this sort of thing.